Thank you everybody for joining us this evening for our first edition of the Young Scientist webinar series for this season. Tonight we'll be hearing from Anna Bolm on Northern California current zooplankton, indiscriminately internalized microplastics. And then our next one is on November 9th. And we will be hearing from Hannah Wellman on using archaeology to understand Native American use of sea otters prior to European contact. A little bit about the Cape Perpetua Cooperative. I should introduce myself too. I'm Tara DuBois. I'm the communications coordinator for the Cape Perpetua Cooperative. It's a pleasure to organize and host this series. And the collaborative's vision is to foster conservation and collaboration within local communities for scientific exchange, management, awareness, and stewardship from the land and sea in and around Cape Perpetua Marine Reserve. And the Marine Reserve is kind of our focal point of the collaborative. And as it's Oregon's largest marine reserve, and it stretches in addition with its marine protected areas from Yachats to Florence. And our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. And you can see the logos at the bottom here. Those are our founding partners, and we've been established for about 4 years now. We do host a variety of community science projects, as you can see here, and you can find all of the events that we host, including presentations and everything that we do. We post on our calendar at capeperpetuacollaborative.org, and then just click the events tab. But upcoming, in addition to this speaker series, we also have our Cape Perpetua winter speaker series that goes through November, Saturdays, November through March. And our upcoming Land Sea Symposium, which is on November 19th. We're still finalizing the agenda, but if you go to the website, you can RSVP and read about our keynote. And I always like to encourage folks to connect with us on our YouTube channel, which is the Cape Perpetua Collaborative. We will be posting a recording of this presentation there, as well as we've got a library of past presentations and some fun videos to add to that as well. And of course, our Facebook page, Cape Perpetua Marine Reserve. It'd be great to see you there. And with that, I'm going to introduce our speaker for this evening, Anna Bolm, who's originally from the Midwest. Anna grew up surrounded by cornfields and never imagined a future studying the ocean. Before joining sciences, she earned a studio arts degree in sculpture from the University of Illinois at Chicago. After a second bachelor's in environmental science and management from Portland State University, she was fortunate to be selected as an Oregon Sea Grant Summer Scholar. And this position exposed her to an exciting variety of marine research at the Hatfield Marine Science Center, where she recently completed her master's in fisheries science. And she is now a phytoplankton ecologist for Oregon State University's NOAA collaboration, the Cooperative Institute for Marine and Ecosystem Resource Studies. And when she's not working, she enjoys freediving, running, foraging, and building furniture. Sounds exciting. And with that, Anna, you can take it over. It's all yours. Thanks again for being with us this evening.
Yeah, thanks so much for having me, and thanks for the introduction. So, and you can see that okay?
Yes, we can see that okay.
Great. So thanks everyone for joining tonight. Like Tara said, my name is Anna Bolm, and I recently graduated with a master's in fisheries science, and my thesis looked at microplastics in zooplankton that were collected off the Oregon and Northern California coasts. So tonight I'm going to share some background information on marine microplastics and what I found, and kind of some ideas on what it could mean. Well, just like I said, just to give you an idea of where we're headed, I'll begin with some background information on marine microplastics, what they are, where they come from, where they go, and why they're a problem. Then I'll share my research for my master's thesis. And lastly, I'll go over what it all means. So we begin. I'll start with a brief explanation for why this study is important. Plastic pollution continues to rise, with millions of tons entering the ocean annually. We know that marine organisms are ingesting microplastics, but are still clarifying the risks, which are hard to assess given their trait diversity. There's also a disconnect between the types of plastics commonly found in the environment from what's used in laboratory studies, limiting the relevance of their results. So currently there are approximately 12.5 to 125 trillion pieces of plastic in the global oceans, and models predict that this amount will triple by 2040. Plastics are sourced from materials designed to last for centuries, making their presence long-lasting and accumulation inevitable. However, microplastics are a relatively new area of research, and therefore we're still working to understand their distribution and effects. Some of you may be familiar with images like this. It becomes pretty obvious how an animal could accidentally ingest plastic or their typical prey item when they look so similar, like we see here with a plastic bag and a jellyfish. But this is a relatively large piece of debris, and we're talking about microplastics, which are much smaller, though this bag will eventually break down into a microplastics. So what exactly are microplastics? There's still debate over how to define them, but for my thesis, I define them as pieces of material that are anthropogenic in origin, between 1 micrometer and 5 millimeters in length or diameter. For reference, 5 millimeters is about the width of a pencil eraser, and a micron is about 1/100th of the diameter of a strand of hair. This size range is fairly common, though some define them as less than 1 millimeter or don't include a lower size limit. And when I say anthropogenic in origin, I'm referring to pieces that appear anthropogenic based on having uniform color and structure, like the fibers in this image. So these fibers were collected about 65 miles offshore during one of our sampling cruises on the Newport hydrographic line, and were identified as polypropylene. Microplastics are often distinguished as primary versus secondary plastics. Primary microplastics are manufactured small, like microbeads in personal care products and abrasives, or nurdles, which are melted down to make larger products, while secondary plastics are remnants from larger plastics, like microfibers from laundered textiles, fragments from degrading debris, or tire dust particles from road wear. A global evaluation by the International Union for Conservation of Nature found that the majority of marine microplastics are from synthetic textiles, car tires, and city dust. To give a quick example, washing a fleece jacket releases about 250,000 fibers into the wastewater, which can eventually make their way to the ocean. As you can imagine, land-based inputs are a major source of marine microplastics. Rivers carry wastewater and stormwater runoff that contains fibers shed from textiles, like that fleece I just mentioned, microbeads from personal care products, and tire dust to the oceans. Microplastics can also enter the ocean as litter from the coastline via wind or runoff following large rain events. Another main source of marine microplastics is the weathering and degradation of larger plastic already in the ocean through photodegradation, physical erosion, or biodegradation. The larger plastic debris can come from mismanaged inland waste or marine activities, including vessel coatings and accidental spills. As the larger items weather in the environment, they break down into increasingly smaller pieces, changing their availability for marine organisms. Once they reach the ocean, the density of the microplastic determines much of its fate. Many plastics, like fibers, foams, films, and low-density fragments, are positively buoyant in seawater and stay near the surface, while the heavier, higher-density plastics settle out to the sediment. However, biofouling and wind turbulence can cause positively buoyant microplastics to undergo a kind of oscillatory vertical migration, mixing the pieces through the ocean's surface wind mix layer. Microplastics can also rise and fall in the water column from ingestion and egestion as they are packaged and repackaged in different forms, changing their density, though eventually most plastics will settle out to the sediment. Global studies show that the distribution of microplastics is both widespread and patchy, with high incidence near more populated areas. The geography of the coastline plays a role in microplastic retention as well. So higher concentrations have been found in protected seas, with concentrations decreasing moving offshore, and overall lower concentrations along open coastlines. Here's a map from a study taken up in British Columbia that highlights the higher concentrations found both near a populated area, Vancouver, and trapped in an inland seas, where light purple is 0 and red is the highest concentration. The numbers are the stations where the samples of seawater were collected. Coastal upwelling can transport buoyant microplastics offshore as Ekman transport moves surface water seaward while deep water rises near shore. A study in a Spanish estuary witnessed this, with higher concentrations of microplastics in surface water sampled collected from the outer estuary than the inner estuary during upwelling conditions. Alternatively, downwelling can transport microplastics onshore when surface waters are transported towards the coast. A study along the Goa coast in India collected 2 times more microplastics from beaches following a period of onshore Ekman transport versus those sampled when currents and winds were moving offshore. Microplastics also ride global ocean currents, which transport them to remote, pristine areas or to convergence zones where they accumulate, like the Great Pacific Garbage Patch in the North Pacific Gyre. Models run in 2018 reported the number of plastics in the Great Pacific Garbage Patch are 4 to 16 times higher than previous reports, and concluded that plastics are accumulating exponentially compared to their surrounding waters. Seasonal variation in microplastic distribution is often linked to oceanographic or weather patterns more than certain times of year. For instance, a study in the Bay of Calvi, Corsica, that sampled surface waters monthly for a year found that wind conditions were more influential than time of year in determining microplastic concentration. Heavy rainfall has also been shown as a significant input for microplastics to aquatic environments. This graph is from a study in the Cooks River estuary in Australia, where they reported a 40-fold increase in microplastic abundance in water sampled 3 days after a storm compared to water from 3 days before a storm. One of the greatest concerns surrounding microplastics is their effect on animals. Their similarity in size to planktonic prey increases the likelihood of them being ingested by a wide variety of marine organisms. Ingesting microplastics has been shown to disrupt feeding behavior, which reduces energy reserves. Microplastic ingestion has also resulted in reduced reproductive success, and in one study, reduced embryonic development in sea urchin. Even if organisms internalize low levels of microplastics from their environment, they may cause harm up the food chain. A recent study found that larval silverside fish who fed on ciliates that had ingested microspheres had significantly lower wet weights than those from the control groups, which suggests the plastics reduced the nutrient intake and therefore growth of the second consumer. This is a map from NOAA's National Center for Environmental Information, which hosts a global repository of microplastic sampling in aquatic ecosystems. The map highlights the differences in sampling efforts between the Atlantic versus the Pacific Ocean, where research is lacking. I mentioned earlier that microplastics are accumulating in convergence zones. However, biological activity is minimal in these areas, but is much higher in the productive waters above the continental shelf. Data is lacking on what concentrations of microplastics are hazardous to marine organisms, leaving the risks to these productive ecosystems poorly understood. I was surprised to find that only a handful of microplastic studies have been performed along the Northern California and Oregon coasts, part of the California current system. Strong upwelling patterns in the California current support a fertile bioregion of resident and migratory marine life that has been called the Serengeti of the sea. Species include Pacific salmon and gray whales, seabirds, and more. This region is both economically important to numerous fishing industries and highly sensitive to environmental stressors like ocean acidification and hypoxia, as well as marine heat waves. Microplastic contamination is an additional stressor that could negatively affect this ecosystem and its associated industries, which is why I wanted to study here. So, onto my research. I chose to study zooplankton because they're an important food source for higher trophic organisms. They often feed in surface waters where less dense microplastics are known to accumulate. And microplastics also overlap with the size range of many zooplankton prey items, putting them at risk for internalization. And their small size and short life cycle make them common laboratory test organisms, ideal for future risk assessments. A major challenge of identifying the risks posed by microplastics is that they have a diverse suite of characteristics. Almost like the uniqueness of a snowflake, each plastic varies in size, shape, color, density, and chemical components, all of which can affect their availability to zooplankton and other marine organisms. Studies have shown that some organisms preferentially feed on select microplastic sizes and shapes, which has been attributed to the animal size or feeding strategy. For example, larger zooplankton can ingest larger microplastics, and suspension feeders may be better suited for larger diameter prey items like fragments versus small diameter fibers. Few laboratory studies have incorporated color as a factor for selectivity, though a field study found a high level of red fibers in this fish species that typically feeds on red algae, which suggests animals could be selecting for color. Laboratory studies with zooplankton have shown that feeding on microplastics can damage feeding appendages, lacerate the digestive tract, and block the passage of food, ultimately limiting food intake. The reduced energy inputs from reduced feeding can have impacts on growth, development, and reproduction. You can see in the video, researchers used microbeads that fluoresce to identify whether the zooplankton are feeding on them. When they are lined through the body, that shows the beads are accumulating. Most impact studies have used virgin microbeads like these when testing the effects of microplastics, which are uncommon in the zooplankton's environment. To appropriately assess any risks posed to zooplankton, it is important to identify the quantities and characteristics of microplastics they internalize in their environment, which can then help inform future lab studies for more applicable results. Field studies off the coasts of Canada, South Carolina, Kenya, Malaysia, and Southeast China have shown that zooplankton are internalizing microplastics in their environment. A study from the Yellow Sea found microplastic concentrations 94 times higher in zooplankton than their surrounding water, suggesting that the zooplankton are retaining internalized microplastics. Other studies have shown that some zooplankton groups internalize more than others, potentially putting some at greater risk. Sampling off the coast of British Columbia, Canada, found that krill had internalized over twice as many microplastics as the copepods analyzed. They attributed this difference to the difference in gape sizes, proposing that larger gape sizes can accumulate more microplastics. After learning about the gap in microplastics data along the Oregon and Northern California coasts, and that we are still trying to identify what concentrations and characteristics are hazardous, I wanted to know whether the zooplankton in this area of the Northern California current are internalizing microplastics, and if so, how many and what kind? Given the patchiness of microplastic distribution, I also wanted to see if there were any changes in internalization between spring and fall, distance from shore, or physical environmental variables. So, I collected plankton toes from 22 stations along the Oregon and Northern California coasts during the spring and fall of 2019. The stations fell along 7 transects perpendicular to the coast between 2 and 372 kilometers from shore. On the map, the samples are shown stacked to see where they were collected for each season, but they were collected from the same station. We used a bongo net like the one shown on the right, which samples over the entire water column, collecting plankton into a cod end at the bottom. Back in the lab, I sorted the tow contents. The picture is of my sorting setup in the hood. 8 zooplankton taxa were chosen for analysis based on groups with high abundance monitored for NOAA's Rapid Zooplankton Assessment. The goal of the Rapid Zooplankton Assessment is to provide timely information to fisheries managers on the zooplankton community composition. The groups that I analyzed included krill, grouped by sizes of greater than or less than 15 millimeters, copepods grouped by size greater than or less than 2 millimeters, amphipods, chaetognaths, crab megalope, and larval fish. I pulled 20 to 50 individuals from each zooplankton group with the exception of larval fish, which I pulled all of due to their low numbers. After separating them by group, shown in the bottom right picture, I examined them under a stereoscope to remove anything stuck to their exteriors. Next, the groups were weighed and transferred to a prepared glass jar for digestion. The separated groups were digested in jars which reduced the organic matter, making it easier to identify microplastics. The digested samples were then vacuum filtered, and the filters were inspected for potential plastics. Here's an example of a filter that has a potential plastic on it. I identified potential plastics as something that appeared anthropogenic in origin due to uniform color and structure and did not break when poked with a dissecting needle, like this piece down here in the corner. The pieces were photographed, measured, and set aside for further analysis. Lastly, I used micro Fourier transform infrared spectroscopy, or micro FTIR, to process a subset of the microplastics representing the diversity of visual characteristics. The micro FTIR helps identify unknown material by passing infrared radiation through a sample to create a spectra of absorbance peaks based on the chemical bond's vibrations. Using the micro FTIR's Omnic software, the readings were matched against libraries of known spectra, like you can see on the right. The top reading is a sample that was matched with the known polyester spectra below. The spectra readings were further verified with OpenSpeccy, an open source spectral analysis software with libraries specific to polymer characterization. First, just to give an idea of what kind of samples I was working with, the zooplankton presence and abundance varied between stations and seasons. I didn't do a full community composition of the zooplankton, but extracted up to 50 from each group when they were abundant enough to do so. The maps are showing the proportion of each zooplankton group at each station by season, and the table shows how many stations each group was present at and the overall number of individuals extracted per season. Overall, 177 samples were analyzed from 20 stations in the spring and 19 in the fall. Both size classes of copepods and chaetognaths were consistently present across stations and seasons, though more large copepods were extracted for analysis in the spring. The other groups weren't always as frequent or abundant, and you can see the crab megalope were only present during the spring at 4 stations. So, to answer my question, are zooplankton in the Northern California current internalizing microplastics? The answer is yes. 82 microplastics were found across all zooplankton taxa analyzed. Here we're looking at how many animals it would take to find 1 microplastic in my samples. For example, I found 1 microplastic for every 12 small copepods, 19 large copepods, and so on. The zooplankton groups are arranged in a rough trophic scale with small copepods at the bottom and chaetognaths at the top, though there was some overlap in the feeding groups. We saw a negative trend with abundance where the more abundant zooplankton had less microplastic internalization. For example, both size classes of copepods and chaetognaths reported the lowest averages for internalized microplastics and were also the most frequently present groups across both seasons. Other studies attributed this negative relationship to a form of biological dilution, where microplastics are less available for a more abundant zooplankton group, from higher competition to feed on the microplastics. However, there is no evidence of true competition And laboratory studies have shown that copepods selectively avoid microplastics when feeding, and even avoid similarly structured algae after ingesting microplastics, which suggests that competition is unlikely. The larval fish average the highest numbers of microplastics. Those larval fish also have the lowest frequency of microplastic occurrence. This suggests that larval fish internalize high levels of microplastics when they are present. They're unable to ingest microplastics as easily as other zooplankton, or they're accumulating more microplastics through trophic transfer. Despite these patterns, I found no significant difference in median amount of microplastics internalized between samples. So now we know about how many microplastics zooplankton internalized, but what kind were they? Only 2 morphologies of microplastics were found. 94% were fibers. Blue fibers were the most abundant color in morphology and were present in all zooplankton taxa. This is consistent with other studies analyzing zooplankton. Blue fibers are also commonly found in seawater samples and are considered a dominant microplastic because of their prevalence in manufacturing and wastewater treatment effluent, particularly from textiles like blue jeans. A recent study found that washing 1 pair of used 100% cotton blue jeans can release 56,000 fibers per wash. Black fibers were the second most frequent microplastic and are commonly reported in other zooplankton studies. It's also worth noting that color identification is subjective, so dark blues and blacks may be categorized differently from different working groups. Fragments were not common in the samples of this study, with a total of 5 internalized by 5 different zooplankton groups. The fragments were found in both seasons and at stations nearshore and offshore, suggesting a minimal but widespread presence. The fibers ranged in size from 0.11 to 3.74 millimeters, with an average of about 1 millimeter. Interestingly, both size classes of copepods internalized the longest fibers. The size of internalized microplastics is thought to vary based on the predator's gape size, feeding strategy, and microplastics availability. A study off the coast in British Columbia reported that on average, krill ingested longer microplastics than the copepods analyzed, and proposed the difference was due to krill having longer feeding appendages, which allow them to consume larger prey. However, fibers have a small diameter and bend or twist, potentially making them more available than more rigid materials, particularly for animals with a smaller gape size. For example, both copepods and krill are known to feed on chain diatoms, which can appear similar to fibers and may explain how they internalize the longest fibers. Overall, there is no difference between the length of internalized fibers across the range of zooplankton taxa sampled, suggesting that fibers are easily confused for food items and are available to multiple feeding strategies and gape sizes. Now onto the results from the micro-FTIR. Of the 82 microplastics isolated, 29 were analyzed. 66% of the materials identified were categorized as non-synthetic, which are in blue on the pie chart. The 2 categories of non-synthetics were cellulose and keratin. The main cellulosic readings were linen, cotton, flax, cardboard, or paper cup. The spectra for cellulose materials are similar and often overlap, so they've been grouped together. Keratin products also have similar spectra readings, so were grouped together, though the top readings were for fur, wool, cashmere, or silk. The synthetic fibers, which are gold on the chart, were made of polyethylene, polyethylene terephthalate, or polypropylene. These materials are all commonly used in textiles, furniture, ropes, sails, and carpets, and can easily enter the environment through laundering or as dust from physical abrasion. My findings were consistent with synthetic fibers internalized by zooplankton in the northern South China Sea, Yellow Sea, East China Sea, and the Bohai Sea. All fragments processed were identified as synthetic. 2 were polyvinyl chloride or PVC, and 1 was polyurethane. Both PVC and polyurethane have many applications. PVC is often used in toys, protective clothing, footwear, and bottles, while polyurethane can be used in packaging, toys, medical appliances, and even cosmetics. Here's an example of a spectra matched in the MicroFTIR's Omnic software. The top spectra is from a blue fiber pulled from a sample of small copepods. The bottom two spectra are the top matches. Here, linen and flax. You can see in the list that cotton, cellulose acetate, and hemp are also included, which have very similar spectra. In summary, yes, all zooplankton taxa sampled from the Northern California current had internalized microplastics. On average, there is no difference between groups in the amount, community composition, or length of internalized fiber. I found mostly blue fibers in my samples, and the fibers ranged from 0.11 to 3.74 millimeters. Fibers are consistently the most common morphology of internalized microplastics in zooplankton studies. Zooplankton often feed in surface waters where low-density microplastics like fibers are known to accumulate, leading them to greater incidence of encounter than higher-density microplastics, which settle out more quickly. The majority of the fibers analyzed were categorized as non-synthetic. This classification is often excluded in microplastic studies as they are not plastics and can be difficult to distinguish from background contamination. Though natural fibers of anthropogenic origin can be just as persistent in the environment and have similar effects when internalized, and therefore were included here. For consistency, I will continue to refer to pieces of anthropogenic material as microplastics, regardless of their potential composition. So, how do we compare to the rest of the world? Compared to similar studies, the averages in my samples from the Northern California current were relatively low. Most of the samples that I analyzed had no evidence of internalized microplastics. A study off the southwest coast of Canada reported microplastic internalization over 3 times higher than this study in the krill and copepods analyzed. Low microplastic internalization is often attributed to low microplastic concentrations in seawater. In studies where both seawater and zooplankton were analyzed, the number of microplastics in the zooplankton was significantly higher than those in the seawater. If a similar pattern holds in the Northern California current, then the low rate of internalized microplastics indicates that the microplastic concentration in seawater is low off the Oregon and Northern California coasts as well. So we also wanted to know if there were any spatial, temporal, or environmental trends. After not seeing any difference among zooplankton groups, the samples were pooled by the site they were collected from. So now we're looking at the average microplastic per individual at each station by season. The values were log transformed for easier visualization, but not for analysis. Looking at the map, we can see that overall internalization was more prevalent in the spring, with presence at 85% of the stations, than in the fall, with 47% presence. We can also see that the 2 stations with the highest microplastic internalization in the spring were the closest stations to shore for their respective transects, and a slight overall trend towards shore in both seasons, though this could be from fewer stations being sampled far offshore. Looking a little deeper, the results showed a weak negative correlation between microplastic internalization and distance from shore for both seasons, which could be from proximity to land and therefore land-based inputs. There were over 6 times as many average microplastics internalized in the spring from the fall, which could be from increased amounts of land-based inputs following winter precipitation. Multiple studies have shown that microplastics contamination increases after high rainfall events, like the study I mentioned earlier in Australia. The increase in microplastics in the environment following rain has also been seen in the resident zooplankton. In China's Bohai Sea, zooplankton had 5 times higher microplastics during the rainy season than those sampled in the dry season. Winters in the Pacific Northwest, as we all know, are notoriously wet. and the increased precipitation could increase the availability of microplastics and therefore internalization among zooplankton. The spring is also an active growth and feeding time for zooplankton, which could explain the increased microplastic internalization. To get an idea of what the sampling conditions were like, here are some graphs of the sea surface temperature, the coastal upwelling transport index, or QD, and the alongshore and cross-shore wind stress for both cruises. The red line indicates the first day of the cruise, and the days following are the averages for each day we were out. During the spring cruise, surface water temperature averaged about 11.5 degrees Celsius versus the fall cruise, which averaged 17 degrees Celsius. The fall samples were collected during a marine heat wave, which could have led to overall lower zooplankton abundances and resultantly lower microplastic internalization. I looked at upwelling and wind stress to see if there were higher microplastic internalization averages near shore during downwelling favorable conditions, but did not see that trend. Overall, the spring cruise had moderate upwelling conditions, and the fall cruise had weak upwelling conditions with variable winds, but no apparent trends with the microplastic internalization. In brief, microplastic internalization had a weak negative correlation with distance from shore for both seasons. There were no clear patterns with upwelling index or wind stress, but the spring samples had 6 times more average microplastics per individual than the fall. I mentioned this could be from increased amounts of microplastics in the water from winter precipitation. However, this is only one sample, so to gain a better understanding, more seasonal studies should be conducted, including seawater samples. I also mentioned that there may be a correlation between increased biological productivity during the spring and microplastic internalization. To identify whether this is occurring, more knowledge is needed on zooplankton community density. So, main takeaways. This study was the first to investigate microplastic internalization by zooplankton off the Oregon and Northern California coasts. I found that major zooplankton taxa are indiscriminate when it comes to internalizing microplastics, with no difference in amount, size, or community composition between the groups sampled. Similar to other studies, the majority of what I found were blue fibers, which are often thought to come from clothing, potentially missed by wastewater treatment plants. Non-synthetic fibers were common, which many observations exclude from their analysis, though lab studies have found they can have similar physical effects as synthetic fibers. And microplastic internalization was 6 times higher in the spring than the fall, which warrants further investigation. So what does this all mean? While the overall levels of microplastics internalized by zooplankton in this study were low when compared to other studies, the input of microplastics to the ocean is persistent with no end in sight. This suggests that the numbers reported here will increase. In particular, a higher level of microplastic internalization following the spring transition could have far-reaching impacts. This period marks the beginning of the upwelling season, when nutrients rise to surface waters, fueling phytoplankton growth and the succession of animals that feed on them. Higher incidence of microplastic internalization during this time could lead to reduced fecundity or energy in zooplankton. Reduced fecundity could eventually reduce food sources, and alternatively, compromised swimming may make zooplankton more available to predators. Also, with the multitude of animals feeding at the same time, the chances of trophic transfer would increase. The waters above the continental shelf are the preferred habitat for multiple listed emigrant juvenile salmon species that feed on krill, larval fish, amphipods, and crab megalopi. The results of this study show that these prey items internalize microplastics. Higher prevalence of microplastic internalization in zooplankton could directly or indirectly threaten an already struggling species and the coastal economies that depend on them. I couldn't have done all this on my own, so a special thanks to Dorothy Horn, Britta Beckler, and Jennifer Fisher for their inspiration, support, and mentorship, which turned a research idea into reality. Thanks to the crew on the Bolm Shimada for safe travels, as well as Sam Zeman, Thomas Murphy, Dave Jacobson, Jess O'Laughlin, Rosie Wood, and Clarice Homer for help with data collection, and thanks to the Brander and Miller Labs, as well as the whole Hatfield community for welcoming me. And thanks to you all for listening. So that's all I have. If anyone has any questions, I'm happy to answer them, or feel free to email me.
Yeah, and if you've got questions, you can feel free to add them to the Q&A section or the chat, and we'll get to them. But I have one I like to ask to kick off.
Sure.
What was the inspiration that inspired you to research this particular topic?
Yeah, so I was already working in a lab at Portland State and working with somebody looking at microplastics. And I think that like waste has just kind of always been something on my mind. I try to be minimal as much as I can, and just thinking about where it goes. So I think it's always been, even in my art, it was kind of a common theme. So working at the lab in Portland State with somebody, and then I had this opportunity to go out on a cruise, and my mentor at Portland State was like, well, if you're going out there, like you have to start collecting samples. So that was just kind of how it started. Yeah.
And then with studies like yours and others similar to it, could they potentially lead to, because you'd mentioned like that wastewater plants or waste treatment plants might be missing things, and potentially even manufacturing companies that manufacture some of these products? Could it potentially affect change?
I mean, that's the hope, you know, is like, I guess, figuring out kind of where it's coming from and like what's most prevalent. And then of course, whether or not like how it's affecting the animals, but you know, there are different ways that wastewater treatment plants can filter the water. And so some of them are better than others, basically. So just narrowing in kind of where the source of the plastics are, I think can help with future management.
Very nice. And it doesn't look like any other questions have rolled in. So I think you had a very clear presentation, it seems. But again, if you, oh, here's one, just a moment. You mentioned the bulk of the microfibers you found were cellulose and keratin. What do you know about the comparing the time of decomposition half-life of these natural products?
That's a really great question. I don't know the actual half-life of the products compared to synthetics. It's definitely, I mean, they break down a lot faster than synthetics do, but it's still not fast enough to where they wouldn't have, you know, any time in the environment. And so I think the idea is that it still persists out there, but, you know, maybe it's for 10 years versus like a couple hundred years. So, I mean, it's good that it eventually breaks down, but, you know, within the time that it's out there, it can still have an impact on different animals.
And let's see, have you encountered studies looking at chemical analysis of seawater looking for the plastic decomposition chemicals?
I'm not sure I understand. Studies looking at chemical analysis of seawater?
Yeah.
So just looking
that look at the plastic from, you know how plastic decomposes? I'm thinking the chemicals from that. Are there studies that look at that analysis of seawater, looking at the chemicals that leach off?
Yeah, that leach off. Yeah, I've definitely read some of those. Actually, what I've seen is that the amount of like leachates that come off of the plastics in the environment, basically animals are taking up just about as much from the water or from the sediment as they would be from plastics. And so as far as like adding more toxicity, it seems like from the studies that I've read, it's not actually adding any more than what's already in the environment, which is also kind of sad and scary.
Yeah. It's always great to learn, but then things are sad.
Yeah. I think that was the question that was being asked.
Any other questions? It doesn't look like it. So with that, Anna, I want to thank you again so much for kicking off the series this year. I look forward to following the research. I always find studies around plastic very interesting. It's such a pervasive thing. But I hope you have a great evening. We've got some thank you Annas in the chat. So with that, and just real quick, I am recording this presentation, so it will be posted on our YouTube channel by tomorrow midday. So if you would like to rewatch it again or share it out with others who might be interested, that's where it'll be available, and you'll get an email with that link. With that, I hope you all have a wonderful evening.
Thanks, everyone.